Okay, so this is the review for Unit 2, Cell Structure and Function. The first objective says distinguish between field of view, focal plane, and depth of field. This is on page 16 of your lab manual that I'm reading from. So before we talk about these parts of the microscope or these characteristics of a microscope, I want to point out a few things here on a microscope. Here are the objectives. You can change the magnification by switching between these objectives. Here's the stage where you put your slide. One way that you can bring the slide in and out of focus is by moving the stage up and down. And the way that you can move the stage up or move the stage down is by turning the coarse adjustment knob here or the fine adjustment knob here. So the three terms that you need to be familiar with about a microscope and how to manipulate them is field of view. This is the area you can see in the microscope. So it's pretty much like the two-dimensional area as well as a three-dimensional area that you can see. So it's everything that you can see when you're looking through that microscope. How can you manipulate this? If you increase the magnification, your field of view decreases. Depth of field, on the other hand, is the thickness of the focal plane. It's how much you can clearly see in the third dimension at the same time. So it's how clear you can see in the three-dimensional space. As you increase the magnification, your depth of field decreases. So notice, the same effect happens for field of view and depth of field. So as you increase the magnification, both of these will decrease. The focal plane is the plane or slice through an object which is in focus under the microscope. So there's only a certain distance from the microscope, from the objectives, that's actually in focus at a time. So this slice can be going through any part of an object. So in order to bring an object into the plane of focus, you have to turn the coarse or fine adjustment knobs, which moves the stage up and down. So let's just look at this. Here we're looking at the field of view. This is the area that I can see. I have a whole bunch of letters on this slide, and if I increase my magnification, I can't see any of the letters that were on the outside. My field of view decreased. We also know that if you increase the magnification, you decrease your depth of field. So if you look here, I'm at a low magnification, 4 times magnification, and this is an image of the stage and the slide. I have 3 threads layered on top of each other. Blue is on top, yellow is in the middle, red is in the middle, and yellow is on the bottom. And as you can see, in the low magnification, all 3 threads are in focus. As I increase my magnification, look what happened. Look at what happens here. So I increase to 10x. Now only 2 of my threads are in focus. My depth of field, the area in the three-dimensional space that's in focus, has decreased. If I increase my magnification even more, so I'm at 40x, now only 1 thread is in focus at a time. My depth of field decreased. Now remember, the focal plane is the slice that is in focus through an object. And so you can see that my focal plane is shown in the yellow here, and only the blue thread is in my focal plane. If I move the stage by turning the fine or coarse adjustment knobs here, I can bring the red thread into the focal plane. If I move it up even further, now I can bring the yellow thread into the focal plane. So I can manipulate the focal plane by moving the stage up and down, by turning the coarse and fine adjustment knob. So the second objective asks you to describe the molecular composition of cell membranes. The cell membrane also sometimes is referred to as a plasma membrane. Really what you just need to know here is that the main molecule that makes up the cell membrane is called phospholipids. So phospholipids are shown in blue in this image here. And just be able to identify those and remember that they are part of the cell membrane. So not a lot of molecules, have to get in and out of the cell, but they can actually fit and squeeze through the phospholipids. So we have these other molecules embedded in the membrane. and these are called proteins. There are special types of proteins in the membrane that actually help transport substances in and out of the cell. All right, so let's move on to objective number 3. It says identify structures and organelles in typical cells of plants and animals and recognize those that are only found in plant cells. So here I have a picture of an animal cell on the left and a plant cell on the right. And let's first talk about what is it that differs between plants and animal cells. Well, if we look over here at the plant cell, we can notice that it has this really large structure here in the middle. This really large structure that you don't see in animal cells is called the large central vacuole. And this is only found in plant cells. Animal cells have vacuoles, these little things here. However, notice they are not as large as what you would see in a plant cell. The purpose of this large vacuole is to create water pressure in a plant cell. That's where vacuoles, their function is to store fluid and nutrients as well as waste. Another structure you can see in plant cells that is not found in the animal cells are these right here. These are called the chloroplast. Chloroplasts are extremely important to plants because this is where photosynthesis occurs. In the chloroplast, you can see these stacks here. These are called grana, or granum for singular. I believe granum is the singular for that. And these are important because these are the areas that actually absorb solar energy on these stacks for photosynthesis. And then the third thing that plants have that animal cells don't have is located right here on the outside. And what is that? That's the cell wall. One mistake that many students make is that they think that plants don't have mitochondria, but plants do have mitochondria. Mitochondria are important for making ATP, and ATP is an energy molecule that both plants and animals need. So both of them have these mitochondria. All right, so we have to be able to identify all these other structures. So as I go through them, I will just tell you what they look like and what they do. So objective 4 asks you to identify the following organelles and know what they do. Now before we talk about those, just wanted to also note that both animals and plants have the cell membrane. Okay, so they do have a membrane covering the whole thing. The plant has the cell membrane, and surrounding that is the cell wall. Okay, so the goal or the role of the cell membrane is to regulate what goes in and out of a cell. You also want to be able to identify the vacuoles, and the large central vacuole is really easy to identify in a plant here, and know what they do. As I said, they store fluid. It's like waste products, nutrients, as well as water. Then we already talked about the chloroplasts, which is located right here. Easy to identify because they have little stacks within them. And what the chloroplast does is it is a site of photosynthesis. This is where glucose is made in plant cells. Next, it says to describe the function of the mitochondria. Well, before I do that, let's talk about how to identify them. They're really easy to identify. Mitochondria looks kind of like a kidney bean, and in the inside it has a very folded second membrane, like that. And the function of the mitochondria is cellular respiration. This is where glucose enters here, the food that you eat, glucose, and what comes out of there is ATP, or energy. Okay, so this is where energy is produced in a cell, and a usable form of energy. So we tend to think of this as a powerhouse of the cell, or where the site of metabolism. Okay, where a lot of metabolic activity happens. All right, so the next one is the Golgi bodies. The Golgi bodies are located right here, as you can see there, also located here. So the Golgi bodies are easy to identify because they look like little sacs like this, layered on top of each other, and then there's little vesicles usually coming out of the sides here. The goal or the role of the Golgi bodies is that lipids that are made in the smooth ER and proteins that are made in the rough ER go to the Golgi, and from there they're packaged and they're modified and then secreted from the cell, or in other words, sent out of the cell. You also want to be able to identify the ribosomes and the endoplasmic reticulums. So ribosomes are all these little dots that you see everywhere. So you see some dots here. I see some dots here in the plant. You also see a lot of the dots attached to this membrane. All of these dots are the ribosomes. Their role is that they're the site of protein synthesis. These little dots actually make the proteins in your cells. And you see these other membranous structures here. The one that has all the dots on it and looks rough is the rough ER. Since it has the ribosomes on it, that means that it also functions in protein synthesis. It packages up the proteins that are made, sends them to the Golgi, where they are then processed and sent out of the cell. Same thing here. Sends it to the Golgi. Those proteins would be processed and then sent out of the cell. The smooth ER, on the other hand, doesn't have any ribosomes attached to it. So you see that down here. The smooth ER doesn't make proteins, but it makes lipids. And then it also will package up those lipids. Those lipids will probably most likely travel to the Golgi. They'll be processed and modified in the Golgi, and then if they are meant to leave the cell, then they will be secreted from the cell. What else did I want to say about that? Sometimes people have a hard time telling the Golgi from the rough and smooth ER. Well, the rough and smooth ER, the membranes are connected to each other. So you're going to see something that looks kind of like this. Okay, so see how all these membranes are connected to each other? And they tend to be close to the nucleus. So these are the ERs. The Golgi, on the other hand, tends to be these sacs, like this, and they are not attached to each other usually in images. So it looks something like that. And you'll see these vesicles starting to form, these sacs starting to form on the outside, where these vesicles bud off to carry things out of the cell. So there might be like a little protein or something in here that's carrying. All right, so that's the endoplasmic reticulums. Last things that you need to know about. In the middle of the cell, we have the nucleus that holds the DNA. Inside the nucleus, we have what's called the nucleolus. It's a little dark stained region. The nucleolus actually synthesizes the ribosomes. So all these little dots out here are made inside here, and then they travel out of the nucleus. So little ribosomes made in there, and then they will travel out. Okay, those are the parts of the cell that you should know. Objective 5 says describe the internal structures of the chloroplast and mitochondria. What you really want to know about this is that both of these internal structures are increasing the surface area to volume ratio. So they're increasing the amount of surface area within these organelles. The chloroplast, again, is a site of protein synthesis. What's really important here is that we have these stacks called grana. These are membranous stacks, and this is where solar energy is absorbed. And then we also have fluid within the chloroplast, and this fluid is called the stroma. Photosynthesis happens both in the grana and the stroma. If we look at the mitochondria, As I said, this is where ATP is made, energy is produced. Energy is actually produced on all these little tiny folds here in the inner membrane, okay? And we call those folds the cristae. And you definitely need to know this term, cristae. No grana are found in the chloroplast. And again, both these structures are going to increase the surface area. So now we have more surface area for ATP to be produced. Also, once again, remember, both plants and animals have mitochondria. Objective number 6 says relate the surface area to volume ratio to cell size and recognize the consequence of this ratio to cell function. So what's really important here is that we're trying to understand why cells are small. Why are cells not really, really large? Well, the reason that this is because of surface area to volume ratio. Or in other words, the amount of surface area compared to the amount of volume. So if we were to look at something that's small, like this, it has a high surface area to volume ratio. The units of surface area are much greater than compared to the units of volume. If you look at something that's really large, so as you increase size, the surface area to volume ratio gets smaller. Now, there's a lot less surface area compared to the amount of volume in that cell. Having a higher surface area to volume ratio is important because it's easier for something to travel into the cell or leave the cell. If I have a very small surface area to volume ratio, it would take forever for these molecules to get to the center of the cell. Cells need to excrete waste and they need to bring in nutrients, and that's really hard to do if you have a very small surface area to volume ratio. So how do cells increase their surface area to volume ratio? They want to maximize that. Maximizing that increases efficiency. One way they can do that is just be smaller. Another way that they could do that is to actually increase the surface area, and you can do that by making folds on the surface area, as you can see here. All these folds increases the amount of surface area. And we saw that in the mitochondria and chloroplasts. Another thing that you could do to increase the surface area to volume ratio is to just increase the number of membranes. If you double the number of membranes, now you've pretty much doubled the surface area and therefore increase that ratio. Okay, that's it for Unit 2. And if you feel good with that, then move on to Unit 3. Now I'm going to go through some practice test questions. And if you feel comfortable with this stuff, then you don't have to watch this. But if you want some more practice, let's go through some questions together. So Jen is looking through a microscope at 3 colored threads. Red on top of blue, blue on top of yellow. She is focused on the blue thread, but the red and yellow threads are blurry. Jen wants to clearly see the yellow thread, so she turns the fine adjustment knob. What did Jen change when she turned this knob? Well, if you remember what I said when we talked about the microscopes, what does turning the fine adjustment knob do? Well, it changes the focal plane. So A is the answer to that question. The next question says, the following figure shows 2 Daphnia under low power magnification. So we're at a low power, only 10 times magnification. If you wanted to decrease the field of view, should you switch to a higher or lower power objective? Well, if you want to decrease or lower the field of view, remember the area that you can see, well, then you're going to want to increase the magnification because remember they have an inverse relationship. So the answer here would be switch to a higher power objective. So go from the 10x to the 40x. Because remember, increasing magnification lowers or decreases the field of view. Next it says, which microscope set up below would have the largest field of view? Well, let's remember it's an inverse relationship again. Well, if we look over here, we first got to figure out what the total magnification is. And this is just telling you when it says 10x ocular, that just means that the eyepiece has 10 times magnification. The objective has 20 times. So to figure out the total magnification, just take 10 times 20, and that's 200. 5 times 4 is 20. 5 times 100 would be 500. And 10 times 4 would be 40. All right, so which would have the largest field of view? Well, the one with the lowest magnification, because they're inverse relationship. So B would be the correct answer here. The next question says, consider the following sequences of change in shape and size for 3 different objects. Which sequence shows a reduction in surface area to volume ratio? So, which of these would show a reduction? Well, we know that when it gets smaller, it increases the surface area to volume ratio. We know that creating folds would increase the amount of surface area compared to the volume. We know that getting bigger decreases the surface area to volume ratio. So A would be correct. And you just got to remember that the larger something gets, the smaller the surface area to volume ratio gets. It's another inverse relationship. The next question asks, which of the following organelles assembles and secretes cell products? Well, we know that this here is the chloroplast. The chloroplast makes glucose, but it doesn't really secrete them from the cell. Secrete means it sends them out of the cell. The mitochondria makes ATP, but doesn't really secrete these cell products from the cell. Here we have the Golgi. The Golgi body actually assembles proteins and lipids, and its main job is to then secrete them from the cell to send them out of the cell. So A would be correct here. The next one says, which organelles have 2 membranes? Well, this is something that we talked about in your worksheet. And we know that both chloroplasts and mitochondria have a double membrane. So D is correct here. What organelle is responsible for storing water in a plant? It's not the cell wall. The cell wall doesn't store the water. The cell wall prevents the cell from bursting. The answer here is the vacuole. The vacuole is the organelle that stores all the water. Let's match some organelles with their appropriate function. Smooth endoplasmic reticulum, what does that do? Well, it synthesizes lipids. The chloroplast is the site of photosynthesis. And the mitochondria, that's where all the ATP or energy is produced, and that is mainly responsible for metabolism, for powering all those chemical reactions happening in your body. So here's one of these type of questions that looks really complex, but it's actually a very, very easy question. So it tells you all this stuff. Imagine that you place several beats in test tubes and all this stuff happens. But look where the main question is here. I'm not even going to read through this whole thing. You can if you would like. But the last sentence says, what kind of molecule forms the pores in this membrane? Well, we only talked about 2 molecules in the membrane, the phospholipids and the actual pores, which are the proteins. So the answer to this question would be proteins. So, still read this whole question, but notice that although this looks really complex, the actual question itself was very simple. Just asking you what creates the pores in the membrane. And I think that is the last, practice question for chapter 2.